Hi all, I am Muhammed and I will be sharing about MoSwA, a tool that we have developed to help better understand viral protein sequence diversity dynamics. I will start with a quick introduction. Viruses, in particular those of RNA genetic material, they mutate rapidly and thus can result in a large number of viral variants. And public databases are treasure troves of sequence data. Given the small genome size of these viruses, they represent an entity with one of the largest numbers of full genome sequences. The ongoing evolution and diversification of viruses poses challenge to vaccine and drug design and their diagnosis and surveillance. Thus, it is crucial to understand the diversity and the dynamics of viral sequence change. We have previously published a methodology for quantitative analysis of sequence change and applied it on viruses such as HIV-1, clade B, and influenza A, H5N1 subtype. Let me elaborate on this one a little. Say we have a protein alignment and we can study the diversity in the context of overlapping chimeras. And let's say we choose a chimer of 9 amino acids because of immunological applications. Each overlapping 9-mer position window in the alignment is then quantitatively analyzed for 4 patterns of diversity motifs. Index is the most prevalent sequence and identified by having the highest incidence. The remaining sequences with at least one amino acid difference from the index are defined as variants and organized into three motifs. First one, the major variant, which is the most common variant sequence, and the next one, minor variant, which are multiple different sequences and each present 2 or more of the aligned viral sequence, and each with an incidence less than major variant. And lastly, unique variants, each observed only once in the given alignment. So this collective incidence of the major, minor, and unique variants at each chimer position, they represent the total variant population for the position. Let's look at the example in here. So let's say I have 20 sequences at this monomer position, and this distinct peptide is found 8 out of 10 8 out of 10 8 out of 20, I'm sorry, 8 out of 20. So this is the index. All others in here are just a variant to the index with an incidence of 12 out of 20. And within these variants, we have this major variant with a frequency of 5 over 20. And then these are unique variants only observed once in this position. Anything in between is a minor variant. And these nonotypes, they are just a measure of the distinct variants at the position. For more information, you can refer to these 2 references from our lab. And in our lab HIV paper, we observed a unique phenomenon, which we termed as index switching. We observed that the dominance of an index was challenged by its variants, where fitness change of 1 or more amino acids, such as through substitutions, change the incidence of a given monomer. So this results in a sequence rank change and thus a motif change. For example, a dominance switch between the major variant and the prevailing index. This can be a little bit too much to digest, so let me illustrate this. So here is the region of an alignment from 310 to 326 with a substitution, with these substitutions. So amino acid E is found 59%, while D is 41%. And as mentioned earlier, the alignment is analyzed using a sliding window of chimer length 9. So where the distinct sequences at each monomer positions are classified into virus diversity motifs. So now let us inspect a monomer position 310 to 318. As you can see in here, The monomer starting with LR, leucine and arginine, and ending with a glutamic acid with E, uh, has the highest frequency and identified as index. It has the frequency of 41%, while the one with the ending with D, aspartic acid, is the major variant and with an incidence of 38%. And the dots in here, they represent the identical amino acids. And in the next position, 311 to 319, although there are slightly changes in the frequency, the motif ranks are maintained till the monomer position of 313 and 320. A motif change happens in the next position. Inspecting this in here, we see that the one with the D now has the highest frequency, which was the previously the second highest and identified as major in here. But right now, it has the highest frequency and it is identified as index. And then in here at the position 317 to 325, a reverse switch happens, restoring the original order. We expected this phenomenon to be also observed between the other motifs and investigated this in our H5 N1 paper, and indeed did witness that this was far more complex than we had imagined, as demonstrated by this illustration here, where all kinds of switches are happening between the different motifs, and we term this broader observation as motif switching. So in order for us to systematically study the motif switching in relation to the viral sequence diversity, we developed MoSwA. It is a command line tool written in Python, and it works both on Windows and Linux system. And it is publicly available through my GitHub page and provided in here. These are some of the unique features in here. I am going to go through each of them separately, so we can skip this part for now. And it should be noted that MoSwA is still in a beta version, and we are hoping for community feedback before we finalize it. First, let me briefly talk about the MoSwA workflow. The input is an MSA file, and with the parameter options to user to define, such as the k-mer size and choice of motifs to analyze, and threshold to tag the low support or highly gapped positions. MoSwA then carries out a quantitative analysis of each k-mer position to define the motifs. This is done by publicly available DEMA library. And MoSwA then takes all the motifs at position P and P1 to compare it and then identify matching k-mers. This process is repeated for all the k-mer positions in the alignment, and motif switch positions are determined and tagged. And MoSwA outputs in total of 10 JSON files if all the motifs were chosen for the analysis, with 2 JSON files per motif, one containing raw results for all the positions, while the second one with the only positions that exhibited motif switch. Additionally, MoSwA produces an HTML report summarizing a short statistical summary on the switches observed and alignment view of all the switches observed in a cluster format, a network graph showing all the dynamic interactions between the motif switches, and a bar plot showing clusters and hotspots of motif switch positions. And also index switch positions are particularly highlighted as positions with possible implications to the vaccine, drug, or diagnostic design. And also a pairwise alignment score for index switches for an indication on the degree of amino acid differences. Here is the sample of the short statistical summary in the HTML report. It provides information such as the input alignment length, and then the average number of the sequences that is analyzed, and also how many positions exhibited low support or no support. And since the position can have more than one motif switch, it also states the number of unique switches out of total switches. And additionally, it is also reported the number of switches are observed for a specific motif. For example, in here, the index, the switches involving in the index is 87, and 220 switches involving in a major variant, among others. The next part of the report is the alignment weave, and index consensus is built by MoSwA to serve as a reference for the alignment and allow for a comparison between the switches. The Kamer positions with switches are marked as asterisks, and while colon represents a motif switch but with a low support. The alignment weave makes it easy to quickly locate all the positions of the motif switches. And this is my favorite part of the tool, a dynamic dot plot of a network graph to show the interactions between the motif switches. The nodes at the top represents different motif switch directions, and the nodes at the bottom, at the top and bottom representing different directions. For example, nodes with a forward trajectory, meaning that higher incidence motif switch to a lower one are grouped at the top in here, while those ones with a reverse trajectory, meaning lower incidence motif switch to a higher one, are located at the bottom. And based on the number of switches, number of edges, we can see the degree of connectivity between the different motifs. We can see that some of them has more, some of them has less, and the color of edges indicating the mechanism behind the generation of switch. For example, gray in here indicates a bona fide switch. We also have pseudo switches where a switch happened because of a motif split or merging, and splits are indicated by blue, while merging ones are indicated in orange. The network graph is dynamic, so one can point out in a node or in a position and see the details of the switch, and you can also click on the legend to inspect only the switches that you are interested in. Also, even more, you can zoom to inspect a specific part of the network. As part of the report, we also provide a summary on any observed clustering of the switches. A cluster is if there are more than 3 switches within the length of Kamer, and clusters that are within a distance of less than 20 amino acids are referred as hotspots. In this example, we can see that number of clusters, and some of them are forming hotspots, as in here, framed in red. And since index switch positions are the most interesting ones, because such positions are possibly to be avoided as vaccine targets given its instability, we produce a similarity spectrum between index and the replacing variant motif based on the PEM30 pairwise alignment. This file is outputted in a tab-separated format, so we can easily open up in any text editor or Excel file. On Excel. In here, in the second column, represents the index that has been replaced, and in parentheses, it is the current motif rank. While in the third column, the sequence is the current index, and in parentheses, it is the motif of the sequence in the previous position. The alignment tells us how the amino acid variability between the sequences switched. A high variability will be indicative of a significant change between the sequences involved in the switch, whereas low one may be less concerning. So in conclusion, motif switching is thought to contribute to the fitness of selection of viral variants. And MoSwA enables a systematic comparative analysis of motif switches within and between viral species proteomes. This can help provide a better understanding of motif switches in relation to viral sequence diversity and evolution, with implications to vaccine, drug design, and diagnosis. And these are the people I want to thank as part of MoSwA team. And thank you for listening to me.